So do you think one of the things that came out early in the development, before we get to targeted therapy, I think it's important for clinicians to remember that there is some literature that would suggest that they have greater sensitivity to pemetrexed-based chemotherapy. So that was an early observation made, I think, by Ross Camage in the Colorado group.
Yeah. And I think that's something that with a rush towards targeted therapy, you bring up a good point that the platinum pemetrexed is what I most often use, and it's generally well tolerated. And you can control the disease for about 8 or 12 months on average with that. And I've sometimes talked to patients like, all right, we've run out of targeted therapies. We need to do what I could describe as a holding therapy, maybe until we can get you onto the next trial. I don't think we should dismiss the role of chemotherapy in this disease.
Well, at some point, we'll get to talking about ALK inhibitors, but at some point you may exhaust your options from an ALK inhibitor, and so don't forget that chemotherapy does play a role in these patients.
I think we're also thinking, that many of these patients live 4, 5, 6 years, and we're really talking about a sequence of therapies rather than just one.
You mentioned briefly the 2 profile trials, so we did have evidence. You know, it's interesting that crizotinib kind of became the standard of care, crizotinib not being necessarily the best ALK inhibitor. It really came into clinical practice as a MET, or clinical trials as a MET inhibitor. But early observations showed responses in ALK, and we had the 2 profile trials, second line, first line, that were clearly better than chemotherapy. So it changed the standard of care. And I think crizotinib was approved in 2011, so just 4 years after the original observation. And it was actually quite dramatic, the benefit, at least in PFS.
It was quite dramatic, and I think the frontline trial sticks more in my mind, where the response rate was 75%, and the PFS was around 11 months. And that was a very good trial in terms of really defining what the benefit is. And also, we haven't touched on this, but the toxicity was better than what chemotherapy offered.
So, if you go back to crizotinib being the standard, then we had the influx of a number of whether you call them true ALK inhibitors or second generation ALK inhibitors. The first one I wanted to kind of discuss is alectinib, because the first trial we saw was the J-ALEX trial at ASCO several years ago, followed again by the ALEX trial that really kind of supplanted crizotinib as the first line therapy. Give me just briefly the top line results of the J-ALEX and ALEX.
So I think importantly, alectinib was really designed to have a narrow focus, more potent ALK inhibition, and what this reduced some of the side effects of it, and also was more active, and it penetrated the blood-brain barrier. And what we saw in both these trials is a dramatic improvement in progression-free survival. If we look at the hazard ratio of crizotinib versus carboplatin pemetrexed, it was 0.5. Of the alectinib versus crizotinib is approximately 0.5, and so we're really improving the progression-free survival. The response rates were slightly higher with alectinib, but it's really the progression-free survival and the lower toxicities that drove, I think, the adoption of this therapy. And then in the ALEX trial, they looked at the patients who developed brain mets, and there was a dramatic reduction in the development of brain mets on the alectinib arm compared to crizotinib arm. And clinically, we really like this idea of sort of preventing brain metastasis rather than treating brain metastasis.
Yeah, so obviously getting to the issue of, can we protect the brain in those patients who don't have brain mets at the time of diagnosis? Do these agents have a protective effect in that setting? I think there's some suggestive evidence to say that that's the case.
I think, they prevented the effect, and I think, interestingly, now we're seeing, I think, more studies looking at small asymptomatic brain mets and then using these therapies as the primary treatment for the brain metastasis, and maybe avoiding or delaying the role of radiation therapy.
So would you agree that based on the J-ALEX and ALEX that alectinib became kind of the standard of care first line?
I think it's unambiguously the standard of care in the first line setting.
I mean, the next drug that I wanted to get your thoughts on is ceritinib. I mean, we had the ASCEND-4 data that compared it in first line to chemotherapy. Give me your thoughts about that trial.
So I think ceritinib showed a lot of promise in the second line setting, but it was dosed at 750 milligrams, and I think
got off to a bad start.
In retrospect, that might have been a higher dose than what was necessary and convenient.
A lot of GI toxicity.
And so I think that was one of the problems. But the ceritinib versus chemotherapy showed a improvement in the progression-free survival. I think the median was about 16 or 17 months on the ceritinib arm, which is impressive. And so it clearly had more potency and activity. They've tried to adjust the dose. The new dose is 450 with food to try to mitigate some of the GI toxicities.
The yeah, and I agree with that. But part of the problem that I've always had with the Ascend-4 trial is that the target wasn't, is it better than chemotherapy? I think crizotinib established that with the profile trials. The question is it better than other ALK inhibitors?
I think they kind of did a trial that was sufficient for regulatory approval, but not clinical adoption. And I think that's what held the drug back. I'm sure there are complex reasons for that, but I think clinically we were much more looking at alectinib versus crizotinib was a much more clinically relevant comparison for us.
Yeah, and then most recently the data with brigatinib. Walk us through the early data. We had a trial comparable to the ALEX trials, comparing brigatinib to crizotinib. Your thoughts?
So I think brigatinib had shown a promising response rate of around 50–60% in the second line, and a median progression-free survival on the FDA-approved dose around 16 months. So it looked like a very active drug. Then it was compared to crizotinib in the frontline setting, treatment-naive patients, at least ALK-TKI treatment-naive patients. And it showed a similar improvement in progression-free survival. On the first interim analysis, hazard ratio around 0.5, and it clearly improved on crizotinib. In the patients with baseline brain metastasis, this benefit was a hazard ratio around 0.2 or 0.3, showing the similar pattern of the CNS activity is really sort of critical in this patient population.
So brigatinib has this unique toxicity, the pulmonary syndrome that occurs early on. Can you kind of walk us through that and what the observation is?
So I think that this is unique to this ALK-TKI, and I think it happens on the frontline trial about 3% of the time. And these are called early onset pulmonary events, and they can show with some dyspnea, some shortness of breath, occasionally some hypoxia. And this is what initiated the dose escalation, where you do 90 milligrams, then escalate up to 180 milligrams after a week, because the patient may develop tolerance to the brigatinib, and then you're less likely to see it in that dose escalation. Clinically, I see these patients back a week after I start the brigatinib, just to make sure that I'm assessing the symptoms and watching them.
Does your staff call them during the day? Because it's usually day 2, 3, 4, or in that range.
We counsel the patients, and then I think we sometimes call them. The pharmacy has a system of calling to make sure that there are not any unexpected side effects.
In the mechanism of action of why this happens is elusive, correct?
Yeah, I've actually asked some prominent people to explain this to me, and I have not really gotten an answer. Brigatinib also hits the EGFR receptor slightly, and that was part of the drug development. And we know that pneumonitis is seen with all these tyrosine kinases. It just seems to be slightly higher with this group, and so it's a unique toxicity.